Okay, so since we're going into the biological approach today, we're going to cover like the contents of biological approach. So we're going to cover the techniques related to it, the localization of function, neuroplasticity, neurotransmitters, hormones and pheromones, genetics and behavior, and finally the HL extension, which is animal research. This is kind of disappointing. Okay. There's no one joining. It's like I'm talking to myself. It's like okay, it's no different to like this. So sad. Okay, so for the techniques used to study the brain in relation to behavior. So generally, like as a scientist or as a researcher in psychology, you need one of the most important factors to determine which technique you're going to use. Depends on your research, like how your research is going to be. First of all, the available technology we have right now, and finally cost, because using these techniques can be very costly, especially if you're doing something like brain imaging techniques. And yeah, different technologies works in different ways to see different things. So some technologies you apply might not necessarily work for other situations. Someone just pops in and pops out. Am I just hallucinating, or is this like really happening? Welp. Okay, so first of all, we have the PET, or the positron emission tomography. So this thing, I forgot how it looks like. This machine shows how the brain activity works, and well, because no one's here and I don't know what is PET, I'm just gonna skip this because this is supposed to be Kat's part, and I have no idea what's happening, and I don't want this give misleading information. So next slide. Oh jeez, wait, I keep on switching back and forth. Okay, there you go. Next up is magnetic resonance imaging. Magnetic resonance imaging is a big magnet that produces 3D images of our soft tissues, like very juicy 3D HD images of them. Basically, how it works is that it uses it has a powerful magnet, right? So that magnet creates a big, strong magnetic field, and in our body we have like these atomic nucleus or protons, and it forces our protons to align with the field. But then after that, the radio frequency is then pulsed through the participant, and after like forcing the protons to align, the protons are stimulated and they spin out of alignment. And like from the spin of the alignment, they have different energies coming out, and they pretty much record that energy and tries to transcribe the energy, so that it can be translated into the brain structure, like very detailedly. Radio frequency, yeah, radio frequency is turned off, and MRI machines measure energy released when protons realign with magnetic field. So there are, yeah, there is 3D images, and there are also usually 2D images. 3D images are usually the ones you see if you've ever seen them, like the interesting, weird, detailed brain shapes. Okay, next up we have fMRI. So fMRI is pretty similar to MRI. Like it's kind of like the same energy, but what it measures is kind of different. If you ever see like those images with the brain with like the strange colors, you're seeing a brain structure that's being measured by fMRI. So how it does it is that it measures the oxygenated blood flow in the brain. Basically, it is believed that the human brain, when it's at its most active, when like your brain is like peak active, the part of the brain that's like working really hard and is like reaching its peak is filling up with a lot of oxygenated blood. So that's what's happening in fMRI. It measures that like oxygenated blood in certain blood areas, and the computer translates those like oxygenated blood rate into a 3D film of the brain activity, and it's mapped in squares called voxels. So usually one voxel equals to 4,000 of neurons. Hi, Yusra. I thought I was speaking alone. I actually I don't know if I'm gonna post like a recording of this because I did a very horrible intro. It was just like me saying hi and then like being disappointed at the turn up rate. Okay, next we have localization of function. So since behaviors in general is like very complex, and it also involves different parts of the brain. Even though there are specific parts that may have specific functions, which we studied in which was shown in some of the studies we learned, well, they still work together with like other parts of the brain to create behaviors. So well, there are also studies that like show that damage to specific parts of the brain causes dramatic loss of the function, but in some cases, retraining is possible to like remap those behaviors into other parts of the brain. But unfortunately, in this session, we're just talking about the theory part of it, so we don't really talk in depth about studies. Okay, so there are three major arguments for localization. There's strict localization. There's specific functions are distributed all over the brain, and there's also relative localization. So strict localization is basically specific parts of the brain have specific functions, and it relates to specific behaviors. So like every specific part of the brain is assigned with like their jobs. If they say specific functions are distributed, they mean like how memory is distributed all over the brain. So there's this argument by some researchers saying that if you take like one portion of the brain, like anywhere, literally, then your memory won't be affected because apparently your brain memory is like distributed evenly. which is an interesting argument. And then last we have relative localization. So it's kind of like the softer stance between both of them. Like you kind of say, oh, some of them are related. Maybe some of them are indeed like that, but then there are some of them that have specific functions, specific brain. Honestly, these presentations are pretty sad. I should have had pictures next time. This looks so plain. Okay, so next up we have neuroplasticity. Neuroplasticity is the ability of brain tissues to take on new functions and build new neural pathways in response to environmental demands. So, basically, how neuroplasticity works is that if you have an environmental stimuli, like your especially a new environmental stimuli, your brain neurons are going to be like, ooh, a new environmental stimuli. So they start creating like they start creating connections between like the synaptic gag and all the neurons and stuff, and they start like branching out. So yeah, imagine like if you have environmental stimuli and then your brain just your brain's like neurons and stuff just starts growing. That's kind of what happens. And when there's that process, that's what you call as dendritic branching. Um, and yeah, areas of the brain can be distributed or gain new functions. And neuroplasticity is most flexible when you're still young, when you're still a child. I think like when you're under 10, that's when neuroplasticity is thriving the most. That's why you see lots of child geniuses that we can't compete with. The more a person does a particular activity, the more neural connections are formed and more neural pathways, physical change in the brain. So that's what you call as long-term potentiation. Okay, but then if you don't practice like a certain behavior, like let's just say when you're young you did ballet, and then when you just stop midway when like you start growing up, your neurons that are associated with like the ballet skills will start to experience this thing called neural pruning, which is basically, well, synaptic pruning, destruction of the neurons, because the brain is tries to be as effective as possible and thinks that, oh, if you have something useless in your brain, it's time to remove it. Okay, this is like a long ass definition of neurotransmission. I, you know what? I'm just gonna skip into the diagram part because it's easier to understand if you know like the diagram. So wait, can I like pointer? Yeah, there you go. That's easier to. Oops. Okay, so um, to remember neurotransmission, it's probably better to try to remember this diagram or like a diagram similar to this and how the diagram works. A neurotransmission is an electrochemical process where an action potential or like a nerve impulse, another name for action potential, but like this side of it, travels along the axon. You see axon right here of a presynaptic neuron towards the axon's terminal. So this is called the axon's terminal. Then when it reaches, you see, after it travels here, there's some these kind of things there. Then it reaches this thing called vesicles. Um, when it reaches this thing, they release neurotransmitters. So the neurotransmitters come out from the vesicles, into the synaptic gap, this area right here, which bind the receptor sites. So these are the receptor sites on the postsynaptic neuron. So this process triggers an axial potential, and the message, like after they go here, the message is sent, and this results in behavior. So the neurotransmitters are then returned to the presynaptic neurons through the transporter sites. So it's kind of like a cycle, except it's like a fancy term cycle that's going on right here. Yeah, if you want to look back at like the long ass explanation later, you can just look back at the presentation. But hopefully that helps you like understand the process better, because I personally, till today, don't fully understand the process of neurotransmission. Well, until I saw this picture. Still hard to grasp, but like I just memorized the terms till there. Okay, so next up we have the types of neurotransmitters. There's the excitatory and inhibitory. I think Kat learned an extra metabotropic neurotransmitter, but well, just keep this as like extra knowledge, I guess. You don't have to memorize it. Just make sure to, well, you have to make sure you have you're memorizing excitatory, inhibitory, agonist, and antagonist. But for metabotropic, maybe you can just add it as like extra info. So excitatory neurotransmitters increases the chance a neuron will fire an axial potential. So it's like acetylcholine, adrenaline, noradrenaline, dopamine, serotonin. So basically it increases like the chance of this cycle happening like in the brain. Inhibitory is the opposite of it. It decreases the likelihood of an axial potential. So it decreases the the chemicals from like sending an electrochemical message. Then for metabotropic, it's like a neutral version, so it doesn't really decrease the likelihood of it. Okay, so other than like the brain part of the neurotransmitters, there's also like neurotransmitters that we try to eat. Wait, no, like eat is like a weird kind of way to describe it. I mean, like you try to consume through pills sometimes, like like the serotonin antidepressant pills, kind of stuff. So there are two types of those. It can be agonist or antagonist. Agonist basically tries to mimic neurotransmitters, tries to increase the neurotransmitters from appearing. Well, antagonist tries to block the receptors. So they try to so like for this part, they try to block it so they don't receive it. And they also try to like remove the neurotransmitters. So they try to reduce like the impact of the neurotransmitters. So there are a lot of types of neurotransmitters, but the more common ones involve acetylcholine, dopamine, serotonin, If I recall correctly, there's many more, but like these are the common ones that they use for studies. Acetylcholine is active in sleep. It can also be responsible for disconnecting the brain from the body, influences memory and cognitive functions, and it also plays a role in memory because there's a study by Roger and Kessners about like acetylcholine in memory, which is so you can use that if you have studies that are using acetylcholine. And then there's dopamine, a stimulant and usually involved in like pleasurable effects. It is also important in learning. Apparently, high levels is linked with schizophrenia, while low levels is linked with the Parkinson's disease. It is also linked with like our behavior in addiction. And then there's serotonin. Serotonin is the workhorse neurotransmitter. It controls thought coherency, mood, sleep, dreaming, appetite. A lack of it is usually associated with irritability, anxiety, depression, and insomnia. However, if I recall correctly, it was like very reductionist to consider to like link serotonin with depression. I'm pretty sure you would have gone more in depth into it if you're part of abnormal psychology. Okay, next up we have neurons. Neurons are the building block of behavior, and it sends electrochemical messages to the brain in response to stimuli. So you don't have to like fully memorize this, because this is like just extra vocab that you need to know. But most of the time, like when you're explaining about neurotransmitters, you should be. Oh wait. Yeah, most of the time when you're explaining about neurotransmission, neuroplasticity, you need to mention about neurons, and yeah, it's the thing that sends electrochemical messages to the brain in response to stimuli. Okay, and then we have neural pruning, which I mentioned before, and long-term potentiation. So when a synapse is not used or underused, it undergoes synaptic pruning, the removal of it. But if a synapse is often used, it undergoes long-term potentiation. So that's basically the strengthening of synapses and neural pathways. Okay, next up we have hormones, pheromones, and their effects on behavior. Hormones. Also before we go into hormones, I like to like interestingly point out that hormones and neurotransmitters are like probably kind of similar to each other in a way, but like not very. Let's see. They're kind of similar, maybe because they interact with each other, but they're different also at the same time. So neurotransmissions are like fast-moving chemicals. Hormones, on the other hand, are slow-moving chemicals, because hormones are released from the specific glands, usually the endocrine system, to the bloodstream, and they regulate medium and long-term changes in the body, like growth, digestion, metabolism, reproduction, like stuff like puberty and hormonal stuff that's happening. That's hormones playing and doing stuff. Since it's like since that's the case, it's hard to voluntarily control. It just happens on its own, and they're only released in places where hormones can bind to a receptor, which is the target cells, or like this specific glands. And it also can lead to a genomic sequence of change. when they bind to those certain areas, which can either cause gene activation or gene suppression. That means that and this is kind of like why it's placed in biological approach, because it changes the genes, so it influences the likelihood of a behavior happening by changing our genes. So it's not a direct effect, it's an indirect effect happening. And it's also why its effects last longer than neurotransmitters, which just happens in like like that fast. It's kind of like if you're comparing it to a computer, your neurotransmitters is your hardware. Your hormones is like when you're trying to load Google Chrome and it takes a while. Then I guess you can compare that as a different. I'm not sure if that's a horrible comparison, but okay. Yes, and there are some hormones that can act as neurotransmitters, like adrenaline, by targeting receptor sites on neuron synaptic act. But you have to take note that hormones and neurotransmitters are perfectly independent from each other. So it's kind of like they have their own systems, but they may interact with each other in order to form certain behaviors. So there is cortisol as one of our hormones. If I don't remember the study name, but one of the studies we have is cortisol. Yeah, because I didn't study that. I didn't study the ones related to cortisol. So, however, it is secreted by adrenal glands, and it helps control blood sugar levels, regulate metabolism, reduce inflammation, assist with or hinder memory formation, the encoding of memory. And it is also released in stressful situations alongside adrenaline. It's also somewhat known associated with the flight fight or flight hormone alongside with the adrenaline. And next we have serotonin, which is known as a contributor to feelings of well-being and happiness. However, like the actual biological function is complex, multifaceted, because it plays a role in cognition, reward learning, memory, and other many more physiological processes, like I explained before. I think one of the studies you can use for this is Crocodile at 2010. I learned that one. And then, if you also have studies for testosterone, you can also use that. Testosterone is produced by the testes in males and adrenal glands for females. It plays a facilitative role in aggressive behavior. Well, according to some studies, it doesn't cause aggression, but higher levels are associated with higher levels of aggression. Then we have oxytocin. If you read the Oxford book, there are lots of studies relating to oxytocin. It's produced by the hypothalamus and secreted by the pituitary gland. I think acting as a neurotransmitter is a mistake. Oxytocin doesn't act as a neurotransmitter. I'm sorry. I'll fix that later. So it plays a role in the mother-child attachment and is believed to play a role in social bonding and trust between people, and it also plays a role in recognition, sexual arousal, trust, and anxiety. And there are studies that shows that it might affect addiction and stress as well. So there's a lot of things going on. Now, in the Oxford study, there's also explanations about oxytocin and trust, oxytocin and ethnocentrism, oxytocin and I forgot the rest, but there are several. Next, we have pheromones. Pheromones, pheromones is interesting but hard. Because the thing is with pheromones is that in humans, most studies that we have is inconclusive. So, however, there's still arguments, like a lot of strong arguments to find this human pheromone because in animals, it's a chemical substance produced and released, which it's produced and released outside, like in the environment, and this chemical that is released outside the environment can help other animals to like detect other animals, like indirectly, as if maybe their gender, mating attraction, mother and child relationship, and etc. So at least in animals, they are in forms of smells that are often undetected. There are two kinds of pheromones in animals, which is the primary pheromones, the slow long-term physiological changes like hormonal effects, and signaling pheromones. So this one usually produce like rapid behavioral effects like mating. You see, in animals, they process pheromones to the VNO or the vomeronasal organ in the nasal cavity. Nasal cavity is usually like kind of like around the nose-ish. I'm not sure exactly where, but it's just around there in animals. And then it's connected to the accessory olfactory bulb. So this accessory olfactory bulb is basically a part of the brain. And this part of the brain, the AOB, is adjacent or also connected to an olfactory bulb, which is another brain region in animals that processes regular smells. So this is why in animals it has something to do with odors, like with smells. However, in humans, it's very hard to say because fetuses, like babies in mother's womb, have AOB, the accessory olfactory bulb, but after birth they disappear for some reason. And not everyone has the vomeronasal organ in humans. Only some of them does, somehow. And even if they do, they're not connected to the nervous system. So like, usually odor smells are like connected to nervous systems because nervous systems are, well, connected to all sorts of things with sense perception, like the nose, the mouth, the touch, etc. But the VNO is not connected to it, so it's kind of weird, right? So they can't really send like the smell signals to the brain and process it like animals. And plus, humans have like over 400, no, not over 400, like around 400 odor receptors, and they all have their own genetic variants. So if you want to really like measure them, you're going to have to test all of them out, kind of, or else it's gonna be hard to figure out what's going on because all of them have gene variants, and all of them, I mean, like every single gene difference can cause differences in the aura, so it's kind of very complicated. Okay, so the pheromones you're going to be covering in your studies would be AND and EST. AND is androstenedione, or if you have struggles remembering it, androstenedione. Although it hasn't reported to significantly affect the mood of heterosexual women and homosexual men. It does not alter or affect behavior overtly. Um, but there are studies associate which are associated with it, and with attention. One of the studies you can use for this is Lundstrom and Olson in 2005 and Hare et al. In 2007. How am I memorizing these studies? Okay, next up we have the EST or S-rate estratenol. I don't know how to say that, but let's just call it S-rate trainol because that's easier to remember. This pheromone, this putative pheromone or hypothesized pheromone is synthesized from androstenedione or androstenedione by aromatase, likely in the ovaries, and it's related to the estrogen sex hormones, but it has no known estrogenic effects. It's first identified in the urine of a pregnant woman. Well, I did not study any of these hormones, but CAT included it, so there's that. Because I studied the major histocompatibility complex, or the MHC. This is basically also a putative or hypothesized human pheromone. It's a group of genes that plays an important role in the immune system. So it is argued that dissimilar MHC gene combinations in parents can produce a stronger immune system in offspring. So basically, because of these genes, it's said that you have different immune system genetic variants. And with that genetic system variant and that like immune system subtle differences, it also helps you create your own unique order, like a specific order formation. And it is argued that if like this is a pheromone, that means that those orders are a form of way for us to like communicate to each other that, oh hey, our genes are different. Like since our order is different, our gene is different, so it will increase our attraction rate, kind of thing that's going on. The study that you can use for this is Redican et al. In 1995. In year 1995. There are also evaluations on that about how like there are issues with the studies and etc. Well, but I'm not gonna go in depth into that unless if someone asks. Okay, now we are going into the relationship between genetics and behavior. So genetics is made out of DNA, and it is inherited from our parents. It helps provide a blueprint for the structure and function of human body. Genome is like all of the genes that an individual process. Genotype is a genetic makeup from parents at birth. Phenotype is when genotype interacts in the environment and like creates a certain trait or characteristic. So let's just say genotype is kind of like let's XXXY in terms of gender. But phenotype is basically saying, oh, XX is females and XY is males, kind of thing. So genotype is kind of like the code. The phenotype is the description of the code. So not all genes that an individual has are necessarily expressed, because some genes can be triggered by outside influences or by inner influences. This is, in other words, this is also called epigenetics. So, how do I describe this curve? It's kind of, I guess it's kind of like when you have like a toy with a set of like its system, and then you try to play with it and interact with it. So like if you do something like this, if you expose this toy into like a certain situation, then it will react differently, kind of thing. Except the brain is more complex than a toy. It's like ultra super complicated toy. Having a gene for a behavior kind of predisposes you to that action, but you don't always show it. So like, yeah, if you have that gene for the behavior, it means that you have a tendency or increased chance that you may have that behavior, but you don't always show it. It depends on the environment or the situation that you were put in. Okay. One of the genes you have is the 5-HT gene. It's the serotonin transporter gene. This influences the reuptake of serotonin, and basically a functional polymorphism in 5-HTT can cause lower or higher transcriptional efficiency. It's usually described as X as lower and L as higher. So S equals lack of serotonin, and lack of serotonin is usually associated with depression. A low serotonin, you know, like how genes are usually paired into two? Yeah. In this case, like a low SS, low ones, two short ones, is more likely to get depression given SLE's GXE interaction. This is according to the stress debt thesis model. So basically, the S allele is more predisposed to depression, while the L allele is not. Then we have the monoamine oxidase oxidase alpha gene, MAOA gene. It's an enzyme that breaks down neurotransmitters in the synapse, so it controls levels of the neurotransmitter. The MAOA gene sends signals from the cells to produce the MAOA enzyme. And the low expression of this gene, MAOL, means that there's less MAOA produced, and that will lead to more neurotransmitters in the synapse. MAOL can increase neurotransmitter levels, like serotonin and dopamine, in important parts of the brain, like the amygdala and prefrontal cortex, while higher levels of serotonin lead to aggressive behavior. Having MAOL gene may encourage aggressive behavior, with less serotonin broken down in the synapse. So basically what it's saying is it's kind of like MAOA is behind the big brains of the workings of serotonin and dopamine, and if MAOL has low expression variant, so yeah, if you have the low expression variant type of MAOAL, that means that MAOAL will control serotonin and make it so that there are increased serotonin in your brain, and it will like decrease the serotonin from going back, and it prefers to let the serotonin stay in the synaptic gap. That's what it's saying. And if you have more serotonin in the synaptic gap, that means that you have like high serotonin, and you have high serotonin, it is usually associated with aggressive behavior. I didn't study this, so I don't know the studies that are related to it, but I've read about MAOI and aggression, so. Okay, next up we have genetic similarities. So, genetic similarities is about relatedness. Basically, in this part of the syllabus, it's argued that the greater the genetic similarities there are, the higher the relatedness. So, they believe that if you have similar genes, you have similar behavior. Because you have like, it's like kind of like having similar toys. Like, yeah, toys with like very similar functions, but with subtle differences. Oh, now that I think about it, it's probably like comparing um, two similar phones with different, like two similar phones in the same set, but different functions. Yeah, probably something like that. So, twin and family studies are useful in investigating the extent of which behavior may be influenced by genetic or environmental factors. Because there's always a very big debate between nurture and nature. Now we go into evolutionary psychology, the last part of the core content before we go into HL content. So, basically, this is backed up by the argument of survival of the fittest by natural selection. And it is assumed that competition for scarce resources like food, partners, can lead to the promotion of favorable traits, whether it be physical, behavioral, or genetic. Well, that's also part of physical, but oh well. The traits enable individuals to acquire sufficient resources to survive and reproduce. So the traits, well, basically, it's kind of like, oh, they found this seemingly beneficial trait, so they're gonna bring it down into each generation, and the trait will survive. Usually, genetics and environmental demands can lead to these kind of evolutionary changes. Okay, now the biological content has ended for the SL. Now we're going into HL, but does anyone have any questions? I assume no one has any questions, but since it's been like almost an hour, I'm gonna give like a 15-minute break. So we're gonna start again at 6:15. Why is my OBS recording the wrong drawbridge window capture? I'm just gonna straight up like not. Yeah, at this point, I'm just gonna straight up not like record it anymore.